Okay, so today we are going to go into, in a little bit of depth, some microbiology that occurred in the 19th century. Microbiology became a discipline in science in the century between 1850 and 1950. And the second half of the 19th century was really when people discovered there were all these microbes out there in the world, and they started identifying them and cataloging them and so forth. But they didn't really put two and two together that these microbes might be responsible for all of the contagious diseases that were well known at the time. You know, things like scarlet fever and typhoid, abscesses, and all kinds of infectious diseases. But it really wasn't recognized that these microbes that were ubiquitous in the environment were really responsible for a lot of the contagious and infectious diseases that were also very well known at the time. And there were two major players in this business that really came to the fore. The first was Robert Koch, who was a young general practitioner physician in northern Germany, who was really working out of his small apartment, where he set up a little laboratory, and he started to culture bacteria that were associated with the diseases of livestock, particularly sheep and cows, and that went by the name of anthrax. Now, you may remember that right around 9/11, there were a couple of letters sent to some dignitaries in Washington with anthrax spores in them, and people died, and a couple of three people in the post office or whatever died. And anthrax was a disease that most physicians had never. I mean, I didn't know much about it. I had to go to the textbooks and start to learn about this whole thing. But in 1876, Robert Koch published a paper about his experiments that he had done with anthrax. And that really was the beginning of microbiology. And at the time, there were other reports that had happened in the years between Jenner and 75 years or so, and Robert Koch. But Robert Koch was really the thing that made people stand up and take notice. And he published a paper in the German literature. It was called The Etiology of Anthrax, based on the life history of the Bacillus anthracus. I say that in the German literature because it had to be translated into English. And at the time, German and the Germans and German science was really at the forefront of things. And if you wanted to be a scientist and you were in the United States, when we were still shooting Indians at that time, out there and so forth, chasing them around, over in Europe, they're a little bit more civilized. But if you were in England or France, you had to also be able to at least read and understand German, and vice versa. The Germans had to understand English and French, but they were still pretty nationalized at the time, and so you just published in your own language. And that's why a lot of the PhD programs in our country, at least at the first half of the 20th century, they had a language requirement, so that you had to show proficiency in a foreign language, and that was the reason. Well, what Koch presented in his paper. Also, one other thing I should say at the time, in the 19th century, if you read the papers that these people published, there weren't really any data. There were just text where the people would describe the experiments, say, well, we did this, I did this, then I did that, and so forth, and then would give you the bottom line, the summary, the interpretation of what happened during the experiments. So there weren't any tables, there weren't any figures, there weren't any hard data that you could read out of these papers and really understand exactly how things were done. Methods were not part of the scientific literature, and results they talked about results, and they talked about the interpretation of the results. And so Koch started in his paper talking about a French worker by the name of Casimir Devane, who in 1850 he found the anthrax bacteria. It was caused by a bacteria as opposed to a virus. And he found them in the blood of diseased animals. And so he could see them under the microscope. They were big enough so that he could see them under the microscope. And by comparison, viruses you couldn't see under the microscope. They were too little. You needed an electron microscope in order to see viruses. He found them in the blood of diseased animals that have ultimately went on to die. And in 1863, he did experiments. This is Devane now, 1863, and he showed that he could transfer the disease using infected blood or contaminated blood from sheep to sheep. And that was sort of the way things sat for quite some time. It wasn't until Koch in 1876 then said, what they knew at the time was that pastures where they had diseased animals and the animals died. And of course, what they would die from, they'd get septicemia, they would get bacteremia. And so they would basically all go into what we call septic shock and die, and that would be it. And then if you left the animals in the fields, in the pastures, and then they deteriorated and whatever, and then if you let that pasture lie fallow for several years, that pasture was suspect, because then if you reintroduced new animals into that pasture, even though there hadn't been anything on it, grazing on it for several years, the disease started up again. So the question was, why was that? And it was just sort of an enigma. Koch says in his paper that Devane was only partially correct in his findings and so forth. And he postulated that these bacteria that formed rods and then chains and long filaments, if you looked under the microscope, they would also form spores, which were little round beads that would form on these long chains of these rods of these bacteria. And ultimately the spores would just sort of fall off those long filaments and just be there. What Koch did was that he used it as his experimental animal rather than the sheep. There also infected cattle and so forth, but he used the mouse. And of course, this is before you could call up Jackson Labs on the phone or Charles River Biological Supply and have them send you 100 mice of all the identical, identical mice and so forth. I don't know where he got his mice, but mice really are great because they reproduce every 3 weeks. They have a litter of 6 or 7 mice every 3 weeks. So it's not too, if you have a colony of mice, you can grow them up pretty rapidly. And now it's the most favorite experimental animal that all scientists know about, because you can do genetic engineering in the mouse and to cells and so forth. And so this is a very good model. What happened was that Koch set up in his laboratory a system so that he could take material that had the anthrax bacilli in it, and he could culture it on his culturing plates that he had. And so that he got a very sort of pure culture that would form colonies on these culturing plates. And he could take a colony and he could inject it into the mouse, and he injected it in the base of the tail. And then he would wait for a while, and he could then sacrifice the mouse, look in the blood and find the bacilli and see them under the microscope, which is what he did. And he devised a microscopic system so that he would take a drop of fluid that had the bacilli in it, and he worked out a system. He could then heat that microscope slide up to 37 degrees and overnight and so forth. And when he did that, he could then look under the microscope at what was there, and this is when he saw these very, very long filaments. And then if you kept on culturing them and kept on looking at them periodically, the filaments would change such that they would get these spores on them, and then the spores would fall off, and you could get then isolated spores and so forth. He then collected all the spores, and he then put only spores into his microscope slides, and he heated it up at body temperature, 37 degrees centigrade, and he found that those spores could change and they could become filaments again. So the filaments would make spores, and the spores that could change back into filaments. And that that was really the critical observation that Koch made, that Devane had not, evidently had not made back in the earlier part of the 19th century. Then what Koch did was that he took only spores and he infected the mice with it, and they got diseased. And he took only filaments, and same thing happened. And the spores were already known by this time to be very resistant to changes in the environment, temperature, either heat or cold, or anything. When the spore would form, it would sort of go into a hibernating phase, but it could then come out of that and be lethal again, and that's what was happening in these pastures. So on the basis of that, in 1876, Koch was noticed by the powers that be in Berlin, and they recruited him to Berlin, and they gave him, and they set him up in a laboratory, and they said, this young man is really, he knows what he's doing. And they set him up in the laboratory, and he started working then in Berlin. And between, the next 10 or 15 years, he gathered together a whole cadre of bacteriologists who ultimately became famous. Their names are famous. The Petri dish was named after Dr. Petri, who was one of Koch's subordinates or progeny. And so then in 1891, the government established the Robert Koch Institute in Paris, or in Berlin, which was really the first research institute that did fundamental research, and it was focused on microbes, microbial kinds of things, primarily bacteria. So that was 1876 when Koch published that paper. And immediately there, almost, in terms of time, thereafter, Louis Pasteur, who was really on the fringes of this whole business for a while, and he had a whole different background and story. In 1878, Louis Pasteur went before the National Academy of Sciences in Paris and gave a presentation about the theory of germs and its application to medicine and surgery. And this was basically a hypothesis paper. But he was, at this stage of his career, he was already a rock star. And he was putting his lance into the ground and establishing the fact that Louis Pasteur was going to go into this business of these microbes. And so he starts off his presentation. I should probably preface that by saying that where he had come from is that he had started out as a chemistry student and did his PhD thesis on organic chemistry at the really the beginnings of organic chemistry in Europe, when people were finding out that there were these organic molecules and so forth. And he made his PhD thesis. He made a major discovery in organic chemistry about different shapes of molecules, isomers. That was really remarkable and stood the test for time. We still talk about these kinds of chimeras that will go on. And then he got into, on the basis of the fact that the shape of the molecule, of the organic molecule, seemed to be what made an organic molecule, as opposed to, which was thought to be only living things could make these kinds of molecules, versus inorganic molecules, or atoms and that sort of thing, and which were thought to be innate and they could not change from inorganic to organic. And the thing that was special about the organic molecules thought to be is that they were living things would make these molecules, and that they would have certain shapes. And this is what he went on to, and thought that he had discovered something that was really fundamental to what life was versus what non-life was. And so he pursued that, and he got into a whole, about 20 years of experiments, trying to basically dissuade the idea of a theory of spontaneous generation. There was a big thing in the mid 18th, 19th century whereby people just thought, God did it. God created this, and life would spontaneously arise out of the primordial soup. Pasteur had gotten into this, and what was really causing fermentation. And on the basis of his interest in fermentation, which was essentially what was going on in creating wine and beer and that sort of thing, is that the yeast, which were single cell organisms, would digest the carbohydrates and form alcohol. And that was alcoholic fermentation. There was also lactic acid fermentation described and so forth. And he worked out the chemistry of this because that's what he was. He was a chemist. But in the process of working all this business out, he got very interested in these microbes doing all these mysterious things in life. And so that by 1878, when he made the presentation to the Academy of Sciences in Paris, he had already shown that the problem in the beer production industry was that the fermentation vats were becoming contaminated with bacteria. Yeast are all right, they make the alcohol, but the bacteria are not so good because they stop that. And he did the same thing with the wine industry, and the same thing with the vinegar industry, and the same thing with silkworms. They were getting infected with bacteria. And so he was doing sort of what we nowadays call applied science to help, all these industries. And of course, in France, you can imagine that the wine and beer industry was really very important. You don't want to screw around with that stuff. It made you feel good. So Pasteur was, and he was already a member of the National Academy. So he stands up in 1878, 2 years after Koch had published this saying that this disease was caused by a microbe. And he said, I've been saying this for several years now to remind you. And he goes on and he says, I have to also remind you that the anthrax bacillus was really discovered by a Frenchman, this guy Devane, and way before Koch got into the game, So the French have been first in so many different kinds of things. And he didn't really say with that much chutzpah, but that's exactly what he had. And you have to go on my website and go on the writing thing and read his papers that he published, or that he published as a result of his presentations to the French Academy, because you get the feeling that this guy, he's really he's competing with Robert Koch. And actually, you have to remember now, 1876, 1878, the Franco-Prussian War was in 1870, and the French lost. And Pasteur did not like the Germans. And actually, they gave him an honorary degree at the University of Bonn. And after the war, he gave it back. He said, I don't want your honorary degree, essentially. And so this created a rivalry between Koch and Pasteur that went on then for the next 20 years, essentially, from 1875 and 1885, 95. So he goes on in his presentation, and he says, the story with smallpox is not just unique to smallpox. People just thought that this whole business with smallpox and the fact that you could get lifelong immunity from the disease if you survived a bout of smallpox, and also you could become vaccinated with the cowpox thing and so forth. That was sort of thought by most people to be a very special case, and they didn't extrapolate that to others. And in this paper, that's what Pasteur did. He said that this is a generalized thing. We know that if you get scarlet fever with streptococci and so forth and so on, and you survive that episode, you don't get that later on, and syphilis and so forth and so on. He goes into all of these infectious diseases. He said that what you really need to do in all of these infectious diseases is you need to be able to isolate the microbe that's associated with the disease. You need to be able to culture that and purify the culture so that there's not any other bugs in that, and then identify that particular microbe in the infectious process. And he said if you can do that, we can probably discover the cause of all of these maladies that are well known to man and God, as well as all of the livestock contagious diseases that have been out there. And that was quite a monumental hypothesis paper that he presented to the Academy. That was in 1878. Well, in 1880, so you can see, here in modern times, if there's one paper that's published in Nature saying X, Y, Z, there's usually two, maybe sometimes three papers that say exactly the same thing. And you say to yourself, why is everybody working on the same thing? Well, it's not that everybody's working on the same thing, but in science, as compared to medicine, science is very, very competitive. And I used to say to my students and postdocs and so forth, is that everybody knows what the questions are that are unanswered. And everybody knows what, that they'd like to get the answer to these particular questions. And usually the problem is it often is reduced to a problem of technical problem that can't be surpassed. And so you come up, you make progress, you run up against a wall, and you can't go any further because there's a technical difficulty that people haven't been able to figure out how to surpass that. In 1880, so between the years of just after Koch published his thing in 76, what Pasteur did was he transformed his total laboratory to microbiology, and he also recruited new people. And he started working on microbes and culturing them and so forth in a big way, big time. And he started studying a disease that was called chicken cholera at the time. And, cholera in the chicken evidently wasn't, a diarrheal disease like we talk about in humans. It was rather a neurological disease. And the chickens would become septicemic, they would get bacteremia, and it would influence their nervous system, and they would sort of go into paralysis and die very quickly. And they died with, teeming with organisms in their blood. And it was a major problem for the poultry industry because they, were losing half of their flocks or even more through this infectious disease. And it would just run rapid through the barnyard. What happened then was that in 1880, Pasteur went before the Academy again, and he then presented a paper entitled "Of Infectious Diseases, Especially Diseases of Chicken Cholera." And so what he did was that he found microbes in the blood of chickens infected with chicken cholera, and he established a method to be able to culture these microbes and passage them from one culture to the next. And he goes through, in his presentation, he goes back through the history and showing that the French were really far ahead of the Germans. He goes back to Devane from 1850 and 1863, and then he states that even though Koch found spores, they were similar to the ones that I found and demonstrated in 65 and 70. So he's really a nationalistic type guy. He restates then, he said, most of these contagious diseases only occur once. He then talks about another Frenchman by the name of Toussaint. And Toussaint was a veterinarian in the Loire Valley who had discovered a microbe in chickens, that all the chickens were infected with this business. And so Pasteur cultured this microbe and passaged it. And he came up with a theory that, they knew at the time that there were some bugs that needed to be in low oxygen or no oxygen in order to grow well, and then other ones that needed oxygen to grow well. And so he thought that maybe he could change the virulence of the chicken cholera bug by culturing it in high oxygen concentrations, or just room air and so forth. And he did that, and he did it over and over and over and over again. And he said he finally ended up with a pure culture of what he thought were attenuated organisms. Attenuation meaning that they no longer were as virulent as the originals that he'd started with. And he showed then that the original bugs that he had were 100% lethal in all the chickens that he would inoculate them into. But these new ones, the Pasteurs over a long time, that were they were attenuated, and he could use that then. He could infect chickens and then wait a while and then infect them with the original cultures, and they would be resistant. And so this is the first time in 1880 where Pasteur indicates that he can take these microbes and change their virulence and make vaccines out of them. And he said at the time that one of the things about these vaccines is you have to use live organisms because his theory about how immunity worked was, and it was based on his observations in microbiology. In microbiology, it became well known during these years that some bugs needed trace nutrients, trace elements. And if you didn't have that in your culture fluid, they wouldn't grow. And so the hypothesis that Pasteur was working under is that when you took these attenuated organisms and you put them into the body, that they would deplete the body of those trace nutrients, so that then if you came back with a virulent sample of that bug, they couldn't grow. That's what he thought immunity was. And he was very adamant about the fact that you really needed to have live organisms to make a vaccine. And it turns out that was very critical. And so basically, what I've always said about Pasteur is that, okay, we know now that live vaccines, bugs that are live but attenuated, are better vaccines than dead ones. But dead ones will work. It's just that the live vaccines are better. And one of the reasons they're better is that you can give a low dose of the vaccine and it will replicate for a while. So you get more bang for the buck. So you put like 10 bugs in and then you get 1,000 out. That's good. You don't have to grow. You don't have to make as much vaccine. And now we're thinking about immunizing 3.5-4 billion people in the world for the coronavirus, and that becomes a major issue. So the reason I mention that is that what he goes on to say in his paper from 1880 is that we can probably do this to every infectious disease. We're going to be able to rid the world of infectious diseases. All we have to do is culture the bugs, attenuate the bugs, and then we can make vaccines for everything. And that's what he said to the National Academy. You have to read his exact words in those papers that are online. I could read them to you, but I won't. They're more fun to read, to live in color and imagine him in front of the Academy. And so I'm going to stop there. We're going to move on the next installment of this journal club into vaccines that Pasteur made for anthrax, and we're going to put the anthrax from Robert Koch together with the vaccine story. So I'll stop there. Thanks. So if you've enjoyed this video, please like, subscribe, and sign up for my newsletter, where I'm serializing my new book, which is called The Quest for New Knowledge. You'll find a sign-up link below. Hey, thanks again. It's been great.